Hello friends, welcome to Dr. Sai Physiology Academy, DOPA for short. This is the place where we'll make the learning of physiology easy, exciting, and effective. Thank you for joining me. And if you're new to this channel, you're especially welcome. And if you love the content that we share, kindly click the like button and also the subscribe button. And don't forget to turn on notifications so that you don't get to miss any new content that will drop. Now let's get started. So today we're going to be dealing with the pancreas, but the endocrine part. We're calling it endocrine pancreas because the pancreas as a gland has both exocrine and then endocrine. Okay, so it's referred to as heterocrine. So it has exocrine and endocrine. So heterocrine. Okay, so it's the exocrine. We've dealt with that in GIT, the part that secretes pancreatic juice for digestive purposes. All right, so and funny enough, it takes up 99%, this exocrine part. So just 1% is what is left for the endocrine part of the pancreas that secretes two major hormones, insulin and glucagon, that we are concerned with. Okay, though it secretes some other thing, we are going to mention them, but we are concerned with insulin and glucagon, which help in the management of blood glucose level. I'm sure you know that already, but let's go a step further. So now you come to the pancreas. Let's just draw a very simplified diagram of pancreas, something like this. And it has all those acini and so on that connects to the second part of the duodenum. Okay, the sphincter of Oddi, ampulla of Vater, and so on. But then you now have what we call islets, little scattered around the pancreas like this, tiny groups of cells. Okay, that's the endocrine part. You see these dots, so we call them the islets of Langerhans. Okay, so it's these islets of Langerhans that make up the endocrine part, which is just 1% of the whole bulk of the pancreas. Now, these islets of Langerhans, they have different types of cells. Okay, you have one, the alpha cells. Okay, the alpha cells secrete glucagon. Glucagon. Then number two, you now have the beta cells that secrete insulin. Then you now have the delta cells. Okay, the delta cells that secrete somatostatin. I'm sure you've heard of somatostatin before. It's also secreted from the hypothalamus. Okay, it's also known as growth hormone inhibiting hormone, but it's also secreted by the delta cells. But we are concerned mainly with this and this. All right, so now in this lecture we are dealing with insulin from the beta cells. So now insulin from the beta cells, the islets of Langerhans, the beta cells are actually more, about 65% of those islets of Langerhans are beta cells, and about 20% is the alpha cells that secrete glucagon. So the beta cells, okay, they make insulin as pre-prohormone. Okay, they are peptide hormones. Remember, we classified different types of hormone. Insulin as a hormone is a peptide hormone. That means it's water soluble, and then it's stored in secretory vesicles. So those islets of Langerhans, they are groups of cells. Okay, so those cells, they secrete, they make the insulin and store them in secret. So they are already there. The active hormone is already there. So whenever the stimulus comes, which of course, high glucose level will stimulate it. We're going to tell you the real mechanism how it stimulates it. Then insulin is released. So now insulin, when it's secreted into the blood, because it's water soluble, it doesn't need to bind to plasma proteins and so on. So it just goes to all the cells of the body, especially muscle cells, okay, muscle cells, fat cells, and so on. But the brain cells, they don't need insulin. All right, so that's one exception. The brain does not need insulin for it to use glucose. Okay, so now what happens is that insulin now has a very short half-life or short duration of action. That means it doesn't last long in the bloodstream because it's water soluble. And a lot of all these tissues in the body, they have what we call insulinase. Okay, that degrades insulin so that it doesn't last so long in the bloodstream and it takes in so much glucose. Because if insulin is present, it will drive all the glucose in the blood into cells, and you don't want that. It causes hypoglycemia. So insulin is the major hypoglycemic hormone. Now we want to talk about the physiological actions of insulin. That's basically what you need to know. But first of all, the mechanism of action. You must remember that insulin is one of those hormones that uses the enzyme linked receptor mechanism. Okay, so the insulin receptor in different cells, they have that intrinsic ability to phosphorylate themselves. Okay, it's called autophosphorylation. So when they are activated, when the insulin binds to its receptor, the receptor, which has intrinsic enzymatic activity, phosphorylates itself and then phosphorylates other proteins within the cell that lead to the physiological actions, right? So that's by the way. So now, how does insulin really perform its physiological actions? Now, the widely known action of insulin is about bringing down blood glucose. But apart from that, insulin is generally known as the hormone of abundance. Okay, so insulin is known as the hormone of what? Abundance. Why is it known as the hormone of abundance? Because its actions generally lead to the storage of metabolic fuels. When you talk about metabolic fuels, I'm talking about proteins, carbohydrates, fats. Okay, storage. Because when you have something in abundance, what do? You put it in a store. When you are eating from hand to mouth, you don't need to store anything. Okay, it's when you have something in abundance. You have 5 bags of rice, you now create a store and store, then you now be using it. So it's a hormone of abundance. So it promotes storage of metabolic fuels. So let's now look at the different effects on the different metabolic fuels. All right, so number 1, what is the effect on proteins? Very simple. Insulin promotes the entrance, okay, the intake of amino acids in cells. Okay, that's simply it. So in this aspect, it does something similar to growth hormone. All right, it does something on this part, this aspect on proteins, promotes amino acid intake. storage. Then number 2, the effect on carbohydrates, which is the very popular one. Let's just write it, carbs. Effect on carbohydrates. Now, it promotes the intake of glucose. Promotes the intake of glucose. And how does it do this? It does it by, when insulin binds to cells, it now through that enzyme link mechanism activates so many enzymes that lead to the insertion of glucose transporter. So glucose transporters now translocate to the cell membrane. So you now have a lot of glucose transport proteins on the cell membrane. So when there are many like that on the cell membrane, they facilitate the intake of glucose. So glucose enters the cell very fast because of insulin. So insulin, that's just simple mechanism. All right. So it's number 1, A, promotes the intake of glucose into cells. Then another aspect also, remember we are talking about that is the hormone of abundance. It promotes the storage of glucose inside there in the form of what? Glycogen. This has to do with especially the liver and muscle cells. Okay, glycogenesis. So the storage of glucose in the form of glycogen, and then also inhibits glycogenolysis, the breakdown of glycogen. It inhibits. So once you understand that it promotes storage, it's easy for you to appreciate all these things that it does. Then also another thing that it does, D, is it also inhibits gluconeogenesis. Okay, I'm sure you know what gluconeogenesis has to do with the formation, okay, of glucose from non-carbohydrate sources, from proteins, from fatty acids. So it inhibits that gluconeogenesis. So all it wants to do is just to store and keep glucose. Doesn't want glucose to be much. Doesn't want glucose to just hang around in the bloodstream. It wants to store it. So this is how it does. It promotes this and so on and so forth. So that's the effect on carbohydrates. Now, lastly, on fatty acids, about the metabolic fuels, fatty acids or lipids. So what does it do? Of course, also it promotes the storage of fats and prevents lipid breakdown. Okay, that means it prevents lipolysis. Let's put it like that. It inhibits lipolysis. You see that? So this is how insulin does everything. It's about storage. It inhibits breakdown of all this metabolic force. It wants them to be stored. That's why it's called hormone of abundance. Unlike its counterpart, glucagon, it's a hormone of starvation. All right. So this is on metabolic fuel. Then insulin has effects on growth. That's interesting. So apart from all these metabolic fuels, it has another effect on growth. Let's write it, number 4, effect on growth. So insulin has a synergistic effect with growth hormone. That's that synergistic relationship. So just as growth hormone promotes growth, insulin also helps it, especially with this, promoting amino acid intake and protein synthesis. So that's how it functions together with growth hormone to promote growth. Remember that growth hormone one of the ways in which it promotes growth, it acts through its second form called insulin-like growth factor. You remember, insulin-like. So insulin promotes growth. All right. So lastly, let's talk about effects on electrolytes. Electrolytes. So insulin also promotes the intake of electrolytes into the cell. And the very common one, popular one, is potassium. Potassium, magnesium, and so on. Potassium especially. In the sense that, when patients in the hospital have what we call hyperkalemia. Hyperkalemia means excess potassium level in the blood. One of the ways in which to drive those potassium into the cells so that they are not high in the bloodstream is by infusing glucose and insulin. That's through drip, okay? An infusion through the veins. You are giving glucose because if you just give only insulin, the person will fall into hypoglycemia, which is bad. So you give glucose and give insulin. As they are working together, potassium, the insulin is driving potassium inside as it's also driving glucose inside the cell. That's how you bring down the high potassium level. So that's what it does. It helps in the intake. It drives potassium and some other electrolytes into the cells. All right. So this is, you see, the physiological actions, very important actions. So the next thing we're going to be talking about is the regulation, and that's where we're going to talk about the real mechanism. How does glucose really stimulate insulin secretion? Don't go anywhere. Very interesting after this break. All right, you're welcome back. So finally, we're going to touch on regulation of insulin. Now, unlike those pituitary hormones, because of the complex things they handle and regulate, they need higher centers, hypothalamus, the brain, and so on. But insulin is not like that. It's just regulating organic nutrient, glucose mainly, and amino acid levels. So it's a very simple negative feedback, which I will explain. Very straightforward. So this is what happens where you need to understand the mechanism in which glucose activates the secretion of insulin, and you see how straightforward it is. Now look at what happens. Now you've taken a meal, and then glucose is produced from your carbohydrate food, enters into the blood, the glucose level now rises. So what happens is that glucose that has entered your bloodstream as a result of the food you've eaten, they now go to the beta cells of the pancreas. And what happens? There's a transporter known as GLUT2. GLUT means glucose transporter 2, type 2. Okay, this mentioning the type 2 is very important, the type, because this glucose transporter type 2 is insulin independent. That's why it's very important that you remember the type. It's insulin independent. That means it doesn't depend on insulin to enter into the cell. Right? It's insulin independent, unlike GLUT4. GLUT4 is what other cells like muscle cells and so on, they use. When insulin now acts, it makes GLUT4 available so much in the cells so that glucose can enter. But this one is a different glucose transporter. Okay, it doesn't depend on insulin. So what happens is that glucose now enters the beta cells through GLUT2, and then through normal process of glycolysis, pyruvate is formed, enters the Krebs cycle, all that biochemistry. Then ATP is produced. It's phosphorylated and ATP is produced. So ATP is produced, of ATP. So what does this ATP do? This ATP now inhibits potassium channels. There are special potassium channels in the beta cells. This ATP has that notorious or special ability to inhibit the potassium channels so that the potassium channel closes. Okay, so this ATP leads to closure of K channels. So what happens when the potassium channel closes? It prevents potassium from going out. You know, potassium usually goes out, likes to go out. So because of that, potassium is positive. It now increases the positivity of the beta cells. So increase in membrane potential. So the increase in membrane potential, what does it do? It now activates voltage gated calcium channels. So there are calcium channels that respond to change in membrane potential. That's why they're called voltage gated. Okay, so this increase closure ATP is produced. Increase in ATP leads to closure of this one, membrane potential. Then this one, membrane potential, now goes to activate calcium voltage gated calcium channels. So calcium now comes into the beta cells, and you know, usually calcium aids exocytosis. It's similar to what happens in nerve cells, the way nerve cells secrete neurotransmitters when they are depolarized. Okay, it leads to calcium entering into the cell and helps in exocytosis. So this increase in the membrane potential, that's depolarization. Let's just put it here. Depolarization. Okay, now activates voltage gated calcium channels. It enters into the cell and it promotes exocytosis of the insulin that has already been stored in secretory vesicle. You know, they are just sitting down there waiting to be activated. So when calcium enters, it activates them and moves them to the cell membrane. Then exocytosis happens. So that's how insulin is secreted. So this one leads to exocytosis, which is the secretion of insulin. So if glucose level is low in the blood, all this, it will not activate all of this. The less ATP will be produced in beta cells, and the less closure of potassium channel, and there is less depolarization, less activation, less exocytosis. So there's that balance. So based on the level of glucose, that's what will determine how much it will activate insulin secretion. So once glucose is high, a lot of insulin is secreted because of how it produces a lot of ATP in these beta cells. That's basically what you need to know about insulin. So in the part 2, we're going to now be talking of glucagon, and that's where we'll now talk of the disorders, especially diabetes. And just for you to remember, there are some substances called incretin that also help to stimulate insulin secretion. They're called incretin. So just read that up there in the text. One of them is called GIP. Another one is called GLP. What does it mean? Glucose dependent insulinotropic polypeptide. Okay, why this one is known as glucagon-like peptide. All right, just for further studies, read the text. You see that they're called incretin. Incretin. They support the secretion of insulin. They help to promote secretion of GIP and GLP. All right, this GIP is also called gastric inhibitory peptide or glucose dependent insulinotropic peptide and glucagon like peptide. All right, so that's where we draw the curtain for today's lecture. See you in the next video.